Good afternoon, everybody. I'm from the University of Helsinki, Faculty of Pharmacy. So I will present the in vitro assay development that we are doing in the context of a SADEM project. So I hope you can see the presentation. So what happens in the fish is the goal that we are trying to study. So the background comes from the environmental risk assessment that is required for the new active pharmaceutical ingredients. That is the regulatory era, that is very heavy process. So in short, it's an exposure-driven two-tiered approach. So in the first phase, if the predicted environmental concentration or the compound's PBT properties exceed a threshold level, then phase 2 experiments are needed. So this involves acute toxicity evaluation in several trophic level organisms, and also the bioaccumulation studies contain the fish experiments. So in principle, the studies are conducted in living organisms. And if we need animal models, so the bioaccumulation study done in fish requires more than 100 fish per API. So the animal-based models are ethically unsound and costly. So if we want to study other compounds that are already on the market, the legacy APIs, for example, or another set of compounds, for example, in the early preclinical drug development phase. So the animal-based models are not the way to proceed. So for these purposes, we develop in vitro tools. So the bioaccumulation of a compound is a dynamic balance between the API intake and elimination. So we can study the drug metabolism also in vitro tools. So in principle, the compounds that are intaken to the fish are metabolically cleared in the liver. And if the metabolic clearance is rapid, it leads to elimination of the compounds, and the toxic risk or biohazard is low. But in the case of compounds that are not metabolically cleared in the liver, or if the enzyme kinetics are slow, then it might lead in bioaccumulation of the compounds into the organisms. Also, another case is the CYP inhibition. So if we inhibit the enzyme activities with a certain compounds, it might affect the metabolic clearance of the other compounds. So these metabolic assays are something that we can study also in vitro. And as a background, the elimination kinetics is difficult to predict by computational tools. So basically, we can use subcellular assays or cell-based in vitro techniques to predict the compound fate. The in vitro tools are ethically sound and inexpensive approaches. And that's why they are more preferred if we need to screen a set of compounds. But these techniques have their shortcomings. We can use primary hepatocytes, but in this case, we also need to extract the cells from living organisms. So we need to sacrifice fish for these cells. The number of animals needed is lower. So in this way, it is more ethically sound than using the whole animals. And also the enzyme activities, for example, in the cells are comparable what happens in vivo. So the in vitro in vivo extrapolation is quite good. Although the cells have a limited lifetime, so we need to extract the cells every time we make the experiments. So it's not fully in line with the 3R principles that we need to reduce the animal use. What comes to the cell lines, they are inexpensive and they can live longer, but they have lost most of their liver-specific phenotypes, so there are limitations in using them as well. If you look at the pictures on the right side, these are on top, they are primary hepatocytes, and then the lower pictures are from cell lines. So in traditional cell cultures, we have the cells adhering on the plastics and they grow as 2D monolayers. But if we inhibit the cell adherence to the plastics, they form spheroids and three-dimensional cultures. So this is one way of improving the viability of primary hepatocytes, but also the cell lines can start expressing the liver enzymes, for example, if they are cultured in 3 dimensions. So in the SUDDM project, we develop in vitro tools to evaluate the hepatic clearance. So in more detail, we develop microfluidic in vitro assays. And why microfluidics? So microfluidics mean that we have a through flow in the assay setup. It advances many of the assay parameters. For example, the lifetime of primary hepatocytes might be enhanced with flow, and also the CYP enzyme expression might be induced with the flow. But in addition, we can design really dynamic assay setups and study more details in more detail, for example, the CYP inhibition mechanisms, if the compound is reversible inhibitor or irreversible inhibitors, and make gradients and such. So it's a versatile way of making the assays. There are challenges, of course, related to these techniques, also relating to the fact that these are developing technologies. So there is no standardization of the protocols. We can use this also as an advantage because we can modify the systems as we wish. We use different polymers. We can tune the polymer properties and, for example, the surface chemistry and functionalize the surfaces. But also, without the standardization, we need to study how the cells react to the material, if the material itself is toxic, and so on. And also, if we use subcellular fractions, the enzyme material interaction should be studied. So in SUDDM project, we developed these microfluidic systems, and then we also are part of the Premier project, which is an IMI project, EU and FPL funded project, which have a lot of industrial partners also involved. So later on, we will also study if these methods that we have developed in the SUDDM project could be further validated for the other purposes. And we will make some feasibility analysis also for the chips that we develop. So some details about the concepts that we are producing. Here is our 3D cell culture chip with microfluidic flow, also called as a liver on a chip. So if you look at the left-hand side pictures, you can see the chip setup. We have the fluidic connections and holders, and in the between we have the chip itself. So it's a straight channel around 3 centimeters long, and at the bottom of the chip we create these micrometer scale microwells to the system. So we load the cells into the system as a single cell suspension, and the microwells work as a trap for the cells. So the cells are sedimented into the wells, and we start the flow in the channel again and rinse off the cells that are not inside the wells, and then we start culturing the cells under flow conditions. So with this system, the surface is made cell repellent. So the cells that are trapped inside the wells will form spheroids and these three-dimensional structures. And we can tune the microvell size, the dimensions, and with this, then we can precisely control how many cells we trapped in each well at the beginning of the experiment. And depending on the cell number, then the 3D growth can be also controlled. And with this, we have possibilities to make versatile characterization of the system. We can control what's going into the chip, and then we can analyze what's coming out from the chip. So, for example, if the cells are metabolizing the compounds that we put into the flow, we can study if the bioaccumulation by evaluating what's coming out from the effluent side. And in addition, we can analyze what's happening to the cells. So we can evaluate the toxicity and different cellular parameters at the same time. And the spheroids can be taken out from the chip, so we can also analyze them in versatile ways. We can use sophisticated microscopic evaluation tools like light sheet microscope, and then we can dissociate the cells from the spheroids and use flow cytometer, for example. So this is one of the concepts that we are developing in Southern project in brief. So the background or the conclusion actually is that if the compounds are bioaccumulating in non-target species, very low environmental concentrations may lead in severe adverse impacts in vivo in different organisms. And we know that the xenobiotic metabolisms and the enzymes that are responsible for drug metabolism in humans, they are widely conserved in different species and across taxonomic groups. So the same enzymes are also found in non-target species. However, the read across from human data is not straightforward because the enzyme kinetics are different and substrate specificities can be different between the species. So when we want to study the bioaccumulation risk,
2 minutes.
Yes, thanks. We have to do it case by case. So we have the in vitro tools available. They are low in cost and low in volumes. But the state-of-the-art methods have their limitations. So in Southern project, we develop microfluidic systems, so completely new tools, and also we try to improve the tools that are already available. So with this respect, we try to develop the versatile and dynamic assays that we could differentiate between the compounds that are rapidly metabolized by the liver enzymes that contains or have a low bioaccumulation risk and low hazard. And then we could differentiate the hazardous compounds and screen or maybe red flag them for further studies.